What's up everybody, it's Photosynthesis Guy. My name is Anthony.
And my name is Gabby. Today we are going to be debunking photosynthesis.
Photosynthesis is the conversion of light energy to chemical energy that is stored in sugars or other organic compounds. It occurs in plants, algae, and certain prokaryotes.
So if you didn't get that, photosynthesis is the process by which green plants and some other organisms use sunlight to synthesize foods from carbon dioxide and water.
Let's break it down.
First, where does photosynthesis happen? In order to answer this question, you need to understand the difference between autotrophs and heterotrophs. Autotrophs are organisms that sustain themselves without eating anything derived from other living beings. They are known as the producers of the environment.
On the other hand, heterotrophs are organisms that sustain themselves by eating other organisms. They are known as the consumers of the environment. Only autotrophs undergo photosynthesis because they must make their own food to survive.
All green parts of a plant, including green stems and unripened fruit, have chloroplasts, but the leaves are the major sites of photosynthesis in plants. Carbon dioxide enters the leaf and the oxygen exits by way of microscopic pores called stomata. Water absorbed by the roots is then delivered to the leaves in veins. Leaves also use veins to export sugar to roots and other non-photosynthetic parts of the plant. Chloroplasts are found mainly in the cells of the mesophyll, the tissue in the interior of the leaf.
Let's take a closer look at the chloroplast. A chloroplast is an envelope of two membranes surrounding a dense fluid called the stroma. Suspended within the stroma is a third membrane system made up of sacs called thylakoids, which segregates the stroma from the thylakoid space inside these sacs. In some places, thylakoid sacs are stacked in columns called grana. Chlorophyll, the green pigment that gives leaves their color, resides in the thylakoid membranes of the chloroplast. It is the light energy absorbed by chlorophyll that drives the synthesis of organic molecules in the chloroplast.
Using molecular formulas, we can summarize the complex series of chemical reactions in photosynthesis with this chemical equation. 6CO2 plus 12H2O plus light energy produces C6H12O6 plus 6O2 plus 6H2O.
The equation for photosynthesis is a deceptively simple summary of a very complex process. Actually, photosynthesis is not a process. These two stages are known as light reactions and the Calvin cycle.
The light reactions are the steps of photosynthesis that convert solar energy to chemical energy. Water is split, providing a source of electrons and protons, and giving off O2 as a byproduct. Light absorbed by chlorophyll drives a transfer of the electrons and hydrogen ions from water to an acceptor called NADP+, where they are temporarily stored. The light reactions use solar power to reduce NADP+ to NADPH by adding a pair of electrons along with the hydrogen ion. The light reactions also generate ATP, using chemiosmosis to power the addition of a phosphate group to ADP, a process called photophosphorylation. Thus, light energy is initially converted to chemical energy in the form of two compounds, NADPH, a source of electrons as reducing power that can be passed along to an electron acceptor, reducing it, and ATP, the versatile energy currency of cells. Notice that the light reactions produce no sugar. That happens in the second stage of photosynthesis, the Calvin cycle.
The Calvin cycle begins by incorporating CO2 from the air into organic molecules already present in the chloroplast. This initial incorporation of carbon into organic compound is known as carbon fixation. The Calvin cycle then reduces the fixed carbon to carbohydrate by adding of electrons. The reducing power is provided by NADPH, which acquired its cargo of electrons in the light reactions. To convert CO2 to carbohydrate, the Calvin cycle also requires chemical energy in the form of ATP. Thus, it is the Calvin cycle that makes sugar. but it can also do so help of the NADPH and ATP produced by the light reactions. Nevertheless, the Calvin cycle in most plants occurs during daylight, for only then can the light reactions provide the NADPH and ATP that the Calvin cycle requires. In essence, the chloroplast uses light energy to make sugar by coordinating the two stages of photosynthesis.
The Calvin cycle can also be referred to as the dark reactions or light-independent reactions, because none of the steps require light directly. Light is a form of electromagnetic energy. The colors we see as visible light include those wavelengths that drive photosynthesis. A pigment absorbs light of specific wavelengths. Chlorophyll A is the main photosynthetic pigment in plants. Other accessory pigments absorb different wavelengths of light and pass the energy on to chlorophyll A. The interaction of light with chloroplast can explain why leaves are green. The chlorophyll molecules of chloroplast absorb violet, blue, and red light, the colors most effective in driving photosynthesis, and reflect or transmit green light. A pigment goes from a ground state to an excited state when a photon of light boosts one of the pigment's electrons to a higher energy orbital. This excited state is unstable. Electrons from isolated pigments tend to fall back to the ground state, giving off heat and/or light.
A photosystem is composed of a reaction center complex surrounded by light harvesting complexes that funnel the energy of photons to the reaction center complex. When a special pair of reaction center chlorophyll A molecules absorb energy, one of its electrons is boosted to a higher energy level and transferred to the primary electron acceptor. Photosystem 2 contains P680 chlorophyll A molecules in the reaction center complex. Photosystem 1 contains P700 molecules. Linear electron flow during the light reaction uses both photosystems and produces NADPH, ATP, and oxygen. Now let's take a look about how the Calvin cycle uses the chemical energy of ATP and NADPH to reduce CO2 to sugar.
The first phase is carbon fixation. The Calvin cycle incorporates each CO2 molecule one at a time by attaching it to a 5-carbon sugar named RUBP. The enzyme that catalyzes this first step is RUBP carboxylase or Rubisco. The product of the reaction is a 6-carbon intermediate so unstable that it immediately splits in half, forming 2 molecules of 3-phosphoglycerate for each carbon dioxide fixed.
Phase 2, reduction. Each molecule of 3-phosphoglycerate receives an additional phosphate group from ATP. Next, a pair of electrons donated from NADPH produces 1,3-biphosphoglycerate, which also loses a phosphate group, becoming 3-GP. Specifically, the electrons from NADPH reduces a carboxyl group of 1,3-biophosphoglycerate to the aldehyde group of G3P, which stores more potential energy. G3P is a sugar, the same 3-carbon sugar formed in glycolysis by the splitting of glucose. For every 3 molecules of CO2 that enter the cycle, there are 6 molecules of G3P formed, but only 1 molecule of this 3-carbon sugar can be counted as a net gain of carbohydrate. The cycle began with 15 carbons worth of carbohydrate in the form of 3 molecules of the 5-carbon sugar RUBP. Now there are 18 carbons worth of carbohydrate in the form of 6 molecules of G3P. 1 molecule exits the cycle to be used by the plant cell, but the other 5 molecules must be recycled to regenerate the 3 molecules of RUBP.
The third phase is the regeneration of the CO2 acceptor, or RUBP. In a complex series of reactions, the carbon skeletons of 5 molecules of G3P are rearranged by the last steps of the Calvin cycle into 3 molecules of RUBP. To accomplish this, the cycle spends 3 more molecules of ATP. The RUBP is now prepared to receive CO2 again, and the cycle continues.
Now that you're here at photosynthesis, pause the video and try to answer the following review questions.
These are the answers in case you had trouble finding these answers.